Hi everybody, I'm just waiting for everyone to join. Hi. Hi Taz. My pleasure, Warren. Hi Tess. Taz, as far as I know, I think it was. But it's nothing to be really worried about because you didn't write it yet. I mean, well, it's not the day of the exam. So you don't need to be concerned. They're going to use the backup paper, if that's what I read correctly on the internet. So don't be too worried. You're just going to write the backup paper. If what I read on the internet was true, and that's a good thing because that means you don't have to come back in December and write a whole new paper that they would then set for you. So don't worry about the paper being leaked. Rather have it earlier than later, just because they're going to use the backup paper tomorrow instead. The backup paper is not more difficult. It's just an alternative, so it probably doesn't. It has exactly the same weighting in it, but we don't need to worry about it being more difficult. It's just a backup paper. It means when the people set the first paper, and it's set by more than one person, those people would have set maybe 5 questions, and then a panel would have chosen which questions to put in each paper. So they have a first paper and a second paper. So I really don't think you guys need to worry about it. Just while I wait a few more seconds for people to join, I have had 2 requests to do something on plant hormones. So I'm going to do a little bit on plant hormones as well. I did the ear in our last live. I'm not really sure if you want me to do something more about the ear this time again. Because I did do balance and I did do posture. I didn't do how we hear though. Yeah. I'm just waiting just a little bit longer before I start. Oh, possible essays. We can talk about that too. Yes, my day has been pretty good. Pretty smooth, pretty good. I can do the path of hearing. No problem. Possible essays. The paper 1 is going to be, paper 1 is probably going to be a nervous system question, or it's going to be a reproduction question. It can't be what it was last year, which was hormones. So it can't be that. All right, I think I'm going to start. Okay, so the request was to quickly recap plant hormones. I can do some homeostasis, no problem as well. We've got time for that. I'm just going to switch the camera around so you can see my page. Okay, so will Quick Sticks do some plant hormones? Okay, so I was going to specifically focus on auxins, right? Because that's where a lot of the questions lie. And yes, Camille, a human impact can be an essay. So I just want to quickly go into auxins. So auxins are our growth inhibitors and stimulants depending on which part of the plant that we are talking about. So generally, what they will give you is there will be a shoot, and essentially auxins are located in the apical tip of the shoot, right? At the very top. And so this happens because what you want to do is you want to have apical dominance. Apical dominance means that you want the tip of the shoot to grow upwards, right? You want it to do that, and you want it to go straight. But what happens if the auxins don't collect all at the top? So this is how it plays out. If light is reaching the shoot in all even directions, in other words, the light is uniform, okay, then you don't need to worry about explaining any changes in the auxins, because uniform light means that the auxins are uniform. However, what happens if light is unilateral and it is coming from one direction? And what that will do is it will cause, if I just draw the original shoot, it will cause all the auxins that were in the tip to collect on the dark side of your shoot. So this is the dark side, this is the light. Okay? Now, if we take that, that's the dark side, that's the light side, and we're receiving light from one direction, okay, then the shoot is going to bend towards the light. And all the auxins are still on this side. Okay, so it bends. But why does it bend and how does it bend? Okay, so essentially in the shoot, and this is really important, the whole time I've only been speaking about the shoot, in other words, it's a part of the stem. The whole time that these auxins collect in the shoot, they are going to effectively prevent our growth from occurring. So what that means is the cells on the dark side, they elongate, which means they get longer. It's almost like rectangles. Whereas the cells on the light side, so the side that's receiving the most light, okay, they do not elongate and they stay small and almost like square-like structures. Now, just to clarify, elongation is when a cell, and I'm just drawing it as a square here, goes from being a small square-like structure to lengthening out and becoming a longer rectangular structure. So auxins in the shoot, they don't like light, so they move away from the light. Okay, so that's what they've done here. They've moved away from the light and they've made these cells elongate and get longer, whereas the cells on the light side are shorter and they don't elongate. That's what causes the bend towards the light. Okay, and this is known as phototropism. And shoots are phototropically positive. It means they're going to draw towards, grow towards the light, okay? But what about shoots? Essentially roots, right? So I'll just swap my page. So often the way that they show a root diagram to you is they don't just draw a root on its own. They will always, in some way or some fashion, attach it to a seed of some kind. So this is my seed. This is the surface of the soil that I've just drawn above here, okay? And they will draw the sort of, this is the shoot, and this is the root. Okay, root coming down. Okay, so let me just label it so you can see it. This is shoot, this is root. Okay, now the shoot. Okay, in the shoot, cells are stimulated by auxins. In other words, if the auxins all concentrate on the side where there is least light, so on this side, it causes bending, which causes it to go up. We've just done that. But what about in the root? In the root, it's going to actually be the opposite. The auxins and roots, they collect on the side that is closest to gravity. Okay? Hi, Nam. So they all collect on the lower side. This is the side closest to gravity. Now, unlike in the shoot, which causes, I'm just going to show you what I mean, which causes the cells to elongate on the auxin side and then not elongate on the other side, on the opposite side. On the root, it's the opposite. Roots don't like auxins, so it actually inhibits their growth, and it causes them to remain inhibited in elongation. So they stay long on the side of the auxins. Short on the side of the auxins, but on the opposite side, they elongate and they get longer, and that's what causes the cells to bend around and grow down. And so if we just pull back away a little bit, you will notice that the auxins are always going to be collecting on the side, if this was underground, on the lower side. Now, here's my little helpful tip to all of you. When you do auxins and you do their impact on growth, you're always going to talk about what happens on the one side, and you're going to talk about what happens on the other side. If you want full marks, that's what you need to do. You need to have a nice rounded answer. You need to say that the cells on the dark side are elongating, whereas the cells on the light side are not elongating. They're being inhibited, which causes it to bend and grow towards the light. For roots, on the other hand, the auxins collect on the side closest to the gravitational pull. Those cells are inhibited and they do not elongate, whereas the cells on the opposite side of the root elongate, and this causes the root to bend downwards towards the ground, and that's what keeps shoots growing up and roots growing down. Okay, there's many different ways that they can ask this, but the one most common question that they always give is, there is a clinostat, which I just want to make sure everyone knows what that is. It's basically a rotating disc. So essentially it looks like a dish, and you turn it sideways and it rotates, okay? And so what they do is they put beans on it in different positions, and the beans start growing, and they say to you, okay, if this continues to rotate, which means it's going around this way, okay, let's assume that this is rotating, my shoot and my root coming out of this, will it continue to grow straight up and straight down, or will it move? Will it change direction? And the answer is it's not going to change direction, everybody, because we are rotating, which means gravity is nice and even. We're keeping the gravity all the way around even, which means everything's going to grow out straight. What about if this wasn't rotating? So let's say this clinostat is not rotating. That then means that our shoot and our root, okay, that's where it's come out the bottom of our seed, if it's not rotating, it will allow the root to turn down and the stem to grow up. But, right, so I'm just going to get rid of this. So now we're not rotating. So this is a picture describing what's happening when we're not rotating. Let me redraw this if we were rotating. So if my clinostat rotates, and I have this, okay, originally they were growing out straight, but if this starts to rotate, my clinostat, doesn't matter if it goes clockwise or anti-clockwise, it's no different. That then means that these are not going to start bending upwards because they can't feel their stimulus because it's being rotated, which means that you'll end up having a shoot that will grow straight and a root that will grow straight, which is not obviously what we want when we plant, plants. We want shoots to go up and we want roots to go down. So this is a clinostat. This is the very common question they give you. The other very common question that they're probably going to give you is, it's like a box. There's an opening in the box. There's a pot plant in there, and there will be some kind of like plant in there, and a light source is coming through here, and it's bending sort of towards the light, like so. And they ask you essentially, how is this portion of the plant blending towards the light with unilateral light? And then they could also ask you, what happens if you put it on a clinostat, which remember I told you is like a rotating disc. It's like a platform that moves. This one is positioned vertically, so it's like standing up on the desk, as opposed to this one, which would be horizontal, and you put the pot plant on top of it and the platform rotates. The moment a clinostat is introduced, everybody, basically you can think of it like this. If you see the word clinostat, you just need to know that the stimulus will be evenly distributed. That's actually the key to this whole entire thing. If you see a clinostat in the question, it basically means that your plant or your bean, your what you call a radical, your growing plant, is not going to experience any stimulus. It's going to be even, whether it's light or gravity. The moment they take the clinostat away, they want you to know that whatever they're experiencing is coming from only one direction. Okay. I'm just going to quickly flip over so you can see me again. So that's the basics to plant hormones. Okay, I can quickly answer this. Tay, what is the difference between the uteral and the menstrual cycle? Okay, so I think what you're meaning is the ovarian and the uteral cycle. So what that means is you need to know the difference between what's happening in the ovaries versus what's happening in the uterus. And it's quite easy to remember because whatever happens in the ovaries affects the uterus. In other words, if the ovaries are secreting estrogen from the follicle, that will influence the uterine lining and make it grow back and grow thicker. And then likewise, if the mother were to, well, I say mother, but if the woman were to fall pregnant, then whatever has happened in the uterus, which would be fertilization and then implantation, that would then influence the ovaries to not make any new eggs, any more follicles, because obviously there's pregnancy. And just before I go on any further, I haven't seen a question a really long time in the final paper, which is a question about the fetus and the placenta. And I feel like they haven't asked a question like that in a really long time. And that makes me think that there's a possibility that they will probably maybe put that in this paper. And it makes me nervous because it's not a common question that they've put in lately. And I feel like a lot of matrics overlook it. There might be a section in your textbook where it's describing, you don't need to know the full fetal development. That's not in the guideline. What you need to know is, how does the placenta attach to the uterine lining, so the endometrium? How does that attach? How do the fetus and the mother exchange nutrients? What vein and what artery does what? And what do the chorion, the amnion, the chorion villi, what do they all do? And I feel that maybe a lot of matrics have overlooked that. And I don't know if it's going to be a question tomorrow, but I feel like it might be. But if it isn't and you haven't gone over that section, maybe you should spend some time doing that today as well. Okay. Just looking for here. Yeah, I know the grade 11s are very lucky. Next year they don't have to write essays anymore, so that's going to be so much nicer for them. I'm just looking for some questions. I think this is Bella asking this. The essay last year was actually about balance and geotropism, so could it be the nervous system again? I'm just thinking now. If they've done balance last year as a part of the essay, the likelihood of them doing it again would not be high. So with that being said, it might be a homeostasis essay tomorrow. And it'll probably be one of those story time homeostasis ones, like someone is running away from something, explain how the body reacts to this, how adrenaline, blah, 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 sweating, vasoconstriction, vasodilation, etc., etc. Yeah. It's either going to be that or it'll be a human reproduction question. I highly doubt they're going to ask you to write out the whole female menstrual cycle. That's normally a question. It's not an essay question. Oh, I can't keep up with all of these. You want me to do the ear? I see lots of ear. And homeostasis, I can do that. You keep an eye on the time. I did meiosis already. I'm sorry, I can't do it. It takes too long, unfortunately. It would take me too long to get all the way through it. Okay, so I'm going to quickly swap my camera around again so you can see what I'm doing. And I'm going to do the ear. I am assuming, though, that you want me to do hearing. I want my diagram in front of me so I can see it while I do this. Let me just find it. Okay. Okay, here we go. So, hearing. Let's quickly do that. Okay. All right, so essentially all you need to know for hearing is the pathway sound takes and how you actually hear. So you have your canal in your ear, right? And at the end of that, you have the tympanic membrane. Attached to the tympanic membrane are the 3 ossicles. And the 3 ossicles have individual names. And so there's our ossicles. Okay. And attached to that is going to be the inner ear. So I'm just going to quickly make a sketch of that. These are my semicircular canals. This is the rest of the utriculus and sacculus. Oh, I haven't drawn this close enough. How am I going to do this now? I think what I'm going to do is, because this is going to be the side, so I'm going to go just in at a circle. Sorry, I'm going to make this as a little bit confusing, but essentially this is the cochlea. Okay, so these are the ossicles. All right, so this is the tympanic membrane, or the eardrum. You can call it whatever you like in the exam tomorrow. Tympanic membrane or the eardrum. These are our ossicles. You do need to know their individual names, so hammer, anvil, stirrup. And essentially what they do is they convert a sound wave, because remember the sound wave comes down the canal. It then vibrates the tympanic membrane. The tympanic membrane then moves each one of these ossicles individually, and it takes a sound and converts it into mechanical movement. So that's what we're doing in the beginning part of the ear. We go from a sound to a mechanical movement. Now the mechanical movement occurs in the ossicles, and they beat against the oval window, and the oval window is the opening into the ear, into the inner ear, which is essentially where sound waves are going to travel into the cochlea, which is this part of the ear down here. This is where we actually hear. And so, one more recap. We take the sound, we make a mechanical movement, these ossicles beat against the oval window. Now, this entire action of going from the tympanic membrane to the ossicle to the oval window is called, so this whole thing of just this section here is called amplification. yes, you will need to include that if this was an essay question or in an explanation. If it asks you what do the ossicles do, the ossicles amplify, and how they do that is you go from a large surface area to a small surface area. So that's what you're doing. You're going from a large membrane to significantly smaller bones, each one is smaller than the next, to the smallest membrane, which is the oval window. Now that then creates pressure waves inside the cochlea. And these pressure waves are in the, you can call it lymph fluid. And the lymph fluid is the fluid that sits inside the cochlea. As this fluid moves, it stimulates the organ of Corti. The organ of Corti then converts pressure waves into electrical impulse, right? Okay. And then from the electrical impulse, you then need to go to the brain, right? And you are going to go to the cerebrum this time because that's where you hear. And essentially, that's how you hear. And so that's what you would need to explain. I'll do it one more time from the top for everybody. So we take a sound wave and we convert it to a mechanical movement. We do this by going from the tympanic membrane through the ossicles to the oval window. And this is where amplification takes place. We move from a large surface area to a smaller surface area. These pressure waves are conducted through the lymph fluid in the cochlea. The cochlea contains the organ of Corti, and the organ of Corti converts these pressure waves into an electrical impulse, and that is then sent to the cerebrum. So that is how we hear. Okay, I'm just going to quickly look at some of your questions because it's quite difficult to read and read all your questions. Let's see if I can go back and see some of them quickly. Let's see. I don't know if there's any questions. I'm trying to find all the questions. I have to scroll so far back. I just also want to say to everybody out there that I really think that you mustn't be too concerned that you're not going to do well tomorrow. I know that you will do well tomorrow, okay? The paper 1 is easier than paper 2, okay? That's notoriously easier for the simple reason that there's more anatomy involved and less sort of abstract topics. I don't know if this is Noni N. She said Kraus corpuscles and Ruffian's corpuscles. You don't need to know any corpuscles names. None. So if you're busy learning about how to sense heat, how to sense cold, you're wasting your time. You could be doing so much other studying. And I tell this to my matrics, which I know some of my matrics are watching now, but you need to study smart. If you're going to be finishing off your studying this evening, you should be spending the majority of tonight, if you are ready for that, doing past papers. That's all you should be doing tonight. If you want to look over some stuff that you're still not good at, then perhaps that's how you should spend your time. But if you are finished studying and revising, then you should spend the majority of your time going over last year's past paper, but also please go over supplementary papers. So those are the people who write in February, March, and June, July. In the previous years, 2019, 2018, there's some really good questions in there, and it is for matrics. I think a lot of people think that that's not for matrics, and that's for people who've already perhaps written matric. Okay, homeostasis, I'm seeing that a lot. Yes, the essay question could be human impact. It was the essay question in 2017. has enough time passed since then for them to do that? Normally, human impact is a case study. It's something like water pollution or air pollution or solid waste removal. It's normally like a situation, a case, and then you deal with it. It's almost like a comprehension. I just want to check on the homeostasis and the endocrine. Genetics. Oh, by the way, I'm going to do this again for paper 2. I know that you write on Monday, so I'll do this on Sunday for everybody who's doing paper 2. And I'll put another TikTok up so you can see me. And just before, if you want to, I will upload this TikTok tonight on YouTube if you want to go back and watch anything. So let me not waste any more time. I'm going to do the homeostasis. So I actually did homeostasis in my previous video, and I've actually made a note on this extensively on how to answer this. But okay, for all your endocrine system, for all of your homeostasis processes, okay, and for actually even your nervous system as well, something like the reflex arc, okay, these 3 things, they all use this structure of answer. I'm going to write the structure of the answer out for you, and you are going to use that to help you remember how to answer any question on regulating anything, okay? So just remember, when we are speaking about homeostasis and endocrine, homeostasis, remember we're talking about the pH, we are talking about temperature, we are talking about water, right? And we're also talking about CO2, but remember CO2 influences the pH. When we talk about endocrine systems, we're talking about the pituitary gland, we are talking about the pancreas, we are talking about the thyroid. And then endocrine also has the ovary and testes in it. We can't forget those as well. And then in the nervous system, you can use these answers for the reflex arc. You can use this for hearing, balance, posture. Okay, so all of these topics that you spend all this time now learning each one of these things and how to answer them, this is my note, and this is how you do it. Okay, this is the template answer. You have to substitute the information in. So this is how it goes, okay? So there is some kind of stimulus. And in your answer, you have to tell me what the stimulus is. If it was the pH changing, you need to tell me it's the pH changing. It's going up or down. If it was the reflex arc, it's because you stood on something sharp or you put your hand on something hot. If it was the pancreas, maybe it's because your glucose levels have gone up or down. There's a stimulus. The stimulus is received by a receptor. Now, you do need to know your receptor names. If we're talking balance and posture, we need to know about the cristae and the macula. If we are talking about water, we're talking about water, we are referring to the hypothalamus, because remember that's responsible for ADH. If we're talking about, I forgot about salt up here too. If we're talking about salt, we are talking about the afferent arterioles in the kidney, which is the receptor. So the receptor receives the stimulus and it makes some kind of corrective measure. What is the corrective measure? Let's use some of our examples from the list above. Let's say, for example, it's the pancreas. This is a common one they love to ask. Let's say the stimulus was there wasn't enough sugar in the bloodstream. So the corrective measure from the receptor, in this case, there's not enough sugar. If it's the pancreas, it is going to be the alpha cells in the pancreas. Their corrective measure is to secrete glucagon. Now that's the corrective measure. That corrective measure needs to be sent to an effector. Now, an effector is generally a muscle or a gland. Okay? And so you need to tell me, where is this substance going to? If I'm still using my glucose one, if this was glucagon, that's the corrective measure, it is sent to the liver. The liver then makes the change. Now, whatever the change may be, perhaps if this was a reflex arc, okay, the reflex arc is when maybe you're pulling your hand away from something that's sharp. The making the change would be the muscles in the hand contracting and pulling away from the sharp object. Now, this is where a lot of matrics stop. You need to keep going. You need to return back to norm. So you need to say that we either say we return to norm or set point. It just means the set point in homeostasis. That's what they refer to. Okay, so I'm going to hold it above so you can sort of see it as a whole. You can use this answer to explain all of these. All you need to do is substitute in what these words represent. So what is the stimulus? What is the receptor's name? What does the receptor's name do? The effector it is sent to, the change that is made, and that we return back to normal. Okay. And that's all homeostasis. Okay, I'm just checking the time. We've got a little bit more time that we can look at something else. If you want to, try to think of something that I would suggest we go over. Because I don't see people asking what I thought that you would all ask, which always concerns me. Whenever my students don't ask me what I think they should be asking me, it makes me worried because they're not asking me questions on the things that matter. They're asking me questions on the things that don't matter. That don't matter as much, maybe. It's Limo. Just so that you know, Limo, I did accommodation and pupillary mechanism in my last TikTok live, which is on my YouTube. So if you want, you can go find the link in my bio and you can watch it there. Are you allowed to use acronyms? Yes, you are, as long as you use the correct acronym. So LH, FSH, estrogen, there's no acronym, progesterone. Trying to think if there's another one. I can't remember or not. Maybe if my matrics are in here, they can answer me this. I can't remember off the top of my head. But I don't know if you need to know interstitial cell stimulating hormone, which is ICSH. But I think essentially you can use any approved scientific acronym. You can use TSH, you can use ADH. Okay, cool. Cool. All right. You know what I'm still worried about? Okay, everyone's asking now what I'm hoping that you were going to ask about, which was about the female negative feedback, because I can't tell you how poorly it is answered, and I don't know what happens to everyone in the exam. Okay, so I'm going to do it for another about 10 more minutes. So I'm going to give you a quick once over for the uterine and ovarian cycle. So let me just flip over on that, and the negative feedback that happens on that. So I did it in my last live, but I'll do it really quick sticks again for you again. So you have two sets of hormones involved in the ovarian and the uterine cycle. So you have the pituitary hormones and you have the ovarian hormones. So the pituitary hormones are your FSH, LH. Ovarian, you have estrogen and you have progesterone. I can't spell now. Progesterone. There we go. Right, so those are our 4 hormones that we are going to need to know tomorrow. Okay, so here we go. In order to start this whole cycle off, we need to start growing a follicle. So that is where follicle stimulating hormone comes in. Follicle stimulating hormone is sent to the ovary, and it produces a follicle, or should I say it takes a follicle and it starts to grow it bigger. So that's follicle stimulating hormone. This follicle, as it grows, secretes estrogen. Now, the purpose of estrogen, if I just write that down here, okay, the purpose of estrogen is it regrows and thickens endometrium. Okay, that's what it's doing. So as that follicle grows, right, we want to prepare our endometrium for implantation. Now. Once that develops, do we need to grow another follicle at the same time? No, we don't. So technically what is happening is, and let me just grab a different color pen so you can see the difference. Essentially what is happening is, as estrogen levels increase, it will have a negative feedback effect on the FSH, which means that as estrogen goes up, FSH will go down. So this is a negative feedback. Okay, why do you say that? Well, because you don't want to be growing another follicle while you're already growing a follicle. Okay, so that means that if I just draw this down here lower, estrogen and FSH have a negative feedback loop. In other words, if one gets too high, the other goes down. If the other one gets too high, then the other one comes back up again. So it's like a seesaw effect, okay? But this happens because we don't want another follicle growing. Now, we've now got this follicle really big, it's mature, it's ready to ovulate. And so how do we release it? Well, we need luteinizing hormone. So luteinizing hormone comes in to our follicle. Our follicle goes through ovulation. And that happens because FSH and LH peak. They become their highest amount. They force the egg out. And you end up with a corpus luteum. Corpus. Oh, I've spelled it wrong. Luteum. There we go. Okay. Now, the corpus luteum, it is really important because it secretes progesterone. So, what is the point of progesterone? Progesterone maintains and makes more vascular. If those of you don't know what vascular means, it means more like veiny. There are more arteries and blood vessels. It's not the same as estrogen. Estrogen regrows and thickens, whereas progesterone maintains and makes it more vascular. And the reason why progesterone maintains it is because progesterone is the indicator of pregnancy. Okay? And so, to round this all off, what would that look like in a graph? Okay, because that's probably how they are going to ask this question. So, I just get a little further away from my page. Here is my axes. Okay, day 0, day 28, roughly day 14 in the middle. Right, let's start off with FSH. So, we want to grow a follicle. So, what happens? FSH increases. Okay. Now, at the same time, estrogen is also going to start increasing, but there's going to be a bit of a lag. They don't increase at exactly the same time. So, this is FSH. This is estrogen. Okay. Now, around day 14, just before that, FSH will take a little bit of a dip, and then it will peak up again around day 14. So it will peak and then it will drop off. Estrogen, on the other hand, is also going to peak around day 14. It'll drop down and then it'll peak again and then drop down. And I'll explain why it peaks twice just now. Let's not forget about our other hormones. Our other hormones being LH up here. So LH, I'm going to put in green. LH is a pretty steady hormone. It sort of doesn't increase until right close to ovulation day, where it increases really high and then it drops off. And literally LH's purpose is to release the egg. That's literally its main purpose. It's just the luteinizing. Luteinizing means to mature. Okay? And then last but not least, if I put in one more color, let's use the purple, we can do progesterone. Okay? And progesterone, also similar to LH, it's sort of just, it's really low for a really long time, past day 14, nothing, and then all of a sudden it peaks. Now, it peaks because we want to know if we are pregnant. Now, if we're not pregnant, it'll drop off. If we are, it'll stay flat. That's what my dotted line is. So this is, it's a very common question, by the way, everybody. How do you know from the graph that someone is pregnant? The clue is that the progesterone levels never flattened, they never decreased. They stayed the same the whole way through. Okay. And that's it in a nutshell, everybody. It really is. I know I've sped through that a little bit. I wanted to tell you why it peaks twice, estrogen. So estrogen peaks twice. It peaks once around day 14 and then once again around day roughly 21. It peaks twice because once, that is where the follicle is at its biggest. So if the follicle is at its biggest, you're going to be making the most estrogen. Okay? And then on day 21, it peaks again because that is the day that a period will roughly start. You can also see it coincides, it's very close to my peak in my progesterone. These two peaks here signify the beginning of a menstruation, so the beginning of a period. So that means that someone would be menstruating between these two points, day 1 and 28. And last little thing that I want to give you as a little tip is they don't always give you the whole graph. They'll give you maybe 2 of these hormones, or they'll give you 1 of them. Or, and this is very important, make sure that they start on day 0. They can actually start on day 14, which means they start the graph like that. And they do that just to make it a little bit more difficult and to make sure that you know what you're doing. Okay. I'm just going to give everyone another maybe 5 minutes to ask me something that I can explain verbally or that they're concerned about. And then I'm going to let everybody go, relax a little bit, study, and get some rest before tomorrow. Okay, Miriam, what is the purpose of FSH? The purpose of FSH is to grow and mature a follicle. Girls are born with follicles. From the moment they are created in the uterus, in their mother's uterus, they have follicles and they stay dormant. FSH takes one of those follicles once a month and essentially matures them and grows them. So that is the purpose of FSH. Takes a follicle, matures the follicle, turns it into a Graafian follicle. No, that's the really big follicle before ovulation. Any last minute tips besides papers? And I saw the other question there, the purpose of LH. The purpose of LH is for ovulation and to finish maturation of the follicle. So LH finishes maturation and it stimulates ovulation. Any more tips for tomorrow? The biggest tips that I can give you is when you get diagram or picture questions, please actually study the diagram. The instruction says that, but I don't think anybody actually does. You look at it, you're like, cool, I've looked at it, now let's go into the questions. Like, unpack the whole thing. If you see that there's like an X and or a Y or a Z, or there's a label A and B, start labeling those things. Make sure you know what you're looking at. Make sure you label everything in that picture before you even answer the questions. And I promise you, it makes a difference. Please take my advice and do that. It's not going to take that much longer. Another tip, please do not write your essay in bullet points. I don't care what your teacher said to you. And I'm saying that very strongly now, but I mark at the end of the year. And I know that we don't like bullet pointed essays. So please don't bullet point your essays. Write full sentences. If you struggle to do that because English is not your first language, then take your bullet points and write them almost like long sentences, okay? Obviously have some full stops in there too, but please don't bullet point down the page, because often you get marks for a couple of terminology that you might leave out if you don't use full sentences. You don't need an introduction to your essay. You don't need a conclusion to the essay either. Those of you who are asking me what the essay topic could be, like I said, they've already done the nervous system and the plant hormones last year, the response to the environment. So it's either going to be a homeostasis question or a reproduction question. Let's see if there's any other questions. There was something else that someone asked just now. No, Mo, if you sleep at 4 a.m., no, that's not good. You need more rest than that. How do you study human impact? You know what I did when I studied human impact? I just made a checklist of all the subheadings out of the textbook that I have. So it would be like, like water quality. And then there's like subheadings in the textbook. And it'll be like water quality. It'll be like wetlands, eutrophication, whatever. And it's just a checklist. So do I know what eutrophication is? Yes. Do I know what, can I explain it? Yes. Do I know about dams? That's water availability. Yes. I literally made a checklist and I studied the checklist. And the checklist is made up of all the subheadings in my textbook. That's how I did it when I was in Matric. And that's how I encourage my Matrics to do it as well. Diagrams they may ask. I've seen so many diagrams, so hard to narrow them down. A neuron diagram, a reflex arc diagram, a brain diagram, an eye diagram, an ear diagram, and a female reproductive system diagram, male reproductive system diagram, and a graph or a cross section to show you what's happening in the female hormones. Those are the most common. So there's about 8 diagrams, and just so that everybody knows, they always use the same diagrams. They never change them. It's always the same ear, the same eye every year. Okay, I think I'm just going to check the time now. I'm going to end the live. Amniotic egg diagram, yes, they can ask that. Let's not forget ovipary, ovovipary, and vivipary. Examples of that. It's also an overlooked one. I know it's a very short section. You know where they normally ask that? In the A, B, both, or none question. Because it's such a small section, they can't put it anywhere else. There or in multiple choice. Difference between meiosis, Jamie Lee, and mitosis and meiosis. So mitosis takes one cell and turns it into 2 identical cells. Meiosis takes one cell and creates 4 non-identical cells, and it halves the chromosome number. That's the main difference. Can the essay be on reproduction in vertebrates? No, because if you look at your exam guideline, there's not enough marks in that section for them to get an essay out of it. They can have like a little paragraph, but that you can't get an essay out of that. Definitely not. What is the pass rate for bio? Well, I feel like the pass rate's always been 30%, which is really low. But I think you mean the pass rate for the paper, not how many people are passing it. I don't know, obviously, that yet. But, yeah. Okay, lots of you are asking me, like, Kelly, do we need to know human impact thoroughly? The ones they always like to do, because we're in Africa, are water availability and quality. So eutrophication, that kind of stuff. Remember when we went through our drought last year and the year before that? That was a nice topic that they included. And then food security and biodiversity. These are like the 3 hot topics that they love to come back to all the time. and they're sort of all interwoven with each other. If you don't have water, you can't grow crops. If you don't have crops and a variety of crops, you reduce biodiversity. Or poaching is also included in decreasing biodiversity. So those are the three main ones they love to ask questions on. The only thing that I would spend a huge amount of time on in human impact, not huge, but I would spend time on, is, can I explain something like eutrophication, which is a process? Can I explain try and think of some of the other solid waste. How do we build those dumps? There's some diagrams in this, in a lot of people's textbooks. There's like diagrams of how to build a solid waste disposal. So many questions. Is dying. How much of plant reactions to the environment? Plant reactions is tiny. It's like maybe 10 marks. It's a very small section. Any other questions before I let you go? No, I think that's it, maybe. Oh, what is the target? It's me. What, miss? What is the target organ for insulin? That's the liver and muscles. You have to write both. Liver and muscles. The ear and eye can be an essay. Yes, I think this is... You do need to know how how a follicle goes from a zygote to a blastocyst. Okay, I think that's all the questions. Yes, I have. I'm sorry, Dylan. I've already done plants. I did it right at the beginning. What topic is for the most marks? It's the nervous system. And then human reproduction. Those are the two biggest sections. And that's normally where the essays are. Which past paper should we go through? Try and go through 17, 18, 19. You don't have to do all of them, obviously. But also maybe look at 2017, 2018, 2019 supplementary exams too. And I am posting this on YouTube now. So the moment we're done here, I'm going to download it off of my phone. And I'm going to put it on my YouTube channel, and you can go look through it this evening if you missed anything from the very beginning. Okay. Julian, I see you asking my essay question. I'm not sure. I still think it's either going to be homeostasis, so response in that way, or some kind of reproduction question. Okay. But I'm going to say goodbye to all of you. And I will post this video up this evening. So have a good study session. Good luck for tomorrow. Bye, everybody.